Remdesivir by Jennifer, Farhad, Annie, and Megan. Hi everyone, this is Annie Moscovian speaking for Team Remdesivir. I just wanted to introduce our group members. Jenny on the top left is a pre-vet student who wants to become a vet one day. Farhad on the bottom left is a pre-pharmacy student who ultimately wants to become a pharmacist. Megan on the bottom right is a marine biology major at Pierce who is working towards a career in research and education. And I'm Annie. I'm a human biology and society major transferring to UCLA in the fall, and I want to become a physician scientist. Together, we present our drug, Remdesivir. So, what is Remdesivir? Remdesivir is an antiviral medication that was invented by Gilead Sciences in July 2015. It actually took Gilead a decade of research before they were able to discover Remdesivir, and they filed for an IND right around the time of the Ebola outbreak in 2015, which is how the drug was first introduced to the public. This medication was originally founded at a drug discovery program at Gilead in order to find inhibitors for the hepatitis C virus. And hepatitis C is actually an RNA virus similar to COVID-19. Remdesivir is currently desirable for the treatment of coronavirus, not because it can cure it, but due to how studies seem to suggest that it may speed up the recovery process by 31%, which gives patients a higher chance of survival. Currently, Gilead has 1.5 million vials of remdesivir that they've produced, which should be enough supply for around 150,000 patients in total. This is the remdesivir timeline. As previously mentioned, it was originally developed for the treatment of viral infections such as hepatitis C. Then in 2015 was used in clinical trial settings in West Africa for Ebola. And in 2020, it's now being used as a potential treatment for COVID-19. Remdesivir remains one of the most promising drugs against COVID, and due to its previous use, it's already passed numerous FDA safety trials. The FDA stated on May 1, 2020, that it is reasonable to believe that the known and potential benefits of remdesivir outweigh its known and potential risks, and it's known to help those who are hospitalized with severe symptoms of COVID-19. It's very important to note that most of the research done on remdesivir has mainly been done in vitro, which means it's outside of the human body, such as in cell cultures and animal models, and it's only just recently been undergoing clinical trials in humans for COVID-19. Additionally, it's important to note that remdesivir can cause an increase in liver enzymes, which means it's potentially damaging to hepatic function and may be evaluated as a risk-benefit analysis when deciding whether to administer to a patient. Again, this doesn't mean that remdesivir is a knockout cure for COVID, but clinical trial data rather shows that hospital stays may be shortened from 15 days to 11 days. And this is thought of as proof of concept, or enough proof to continue with the drug because it shows some improvement, even if not all. However, other studies out of China and even in the US have conflicting evidence of its efficacy, and it's still under review. However, until other methods of the treatment, such as vaccination, are discovered, remdesivir will be continued to be rolled out in hospital settings. As of May 14th, all US states have received some amounts of remdesivir, but not nearly enough. The California Department of Public Health has enough medication for around 100 to 200 patients, depending on how long they need to take the drug. This state has more than 3,000 patients in the hospital with coronavirus. San Francisco, for example, has enough doses for only 3 to 5 patients, but the city currently has about 70 patients hospitalized with COVID-19. So the main problem is getting enough remdesivir to enough patients in time.
Scientists are currently reviewing numerous targets to stop the novel coronavirus, including viral entry, cleavage, different stages of translation, viral release, as well as RNA replication, which is where remdesivir is key. Remdesivir belongs to one of the oldest classes of antiviral drugs, nucleoside analogs, that target the polymerase enzyme that replicates RNA, the virus's genetic material inside the host cell. Viral information is replicated through RNA structures once it takes over the cell of the host and its replication machinery. The quick flashback to biology class shows the single-strand structure of RNA and its building blocks. The base is uracil, guanine, adenine, and cytosine that together produce sequences of genes that build into creating proteins and ultimately the genetic code for viruses like COVID-19. Here we see the structure of the RNA base adenosine, and here the structure of remdesivir, for a side-by-side comparison of the similarities of their structures in functional groups. It is because of the similarity in structure that remdesivir is able to present itself as one of the novel coronavirus's RNA building blocks. As a nucleoside chemical, it mimics the structure of a natural nucleotide, adenosine, and may be inserted by the RNA polymerase, grabbing remdesivir instead of the actual base and inserting it into the novel coronavirus's genetic code, causing a structural modification that stops the replication process. This important structural difference is noted in the highlighted number 4, a 1' cyno group that has been shown in lab settings to be a very effective inhibitor of viral RNA polymerases. This interruption stops the virus from multiplying and infecting more cells in the body and induces an irreversible delayed chain termination, meaning once it plugs into the RNA structure, the chain is terminated and not grown to completion, with remdesivir's presence causing a delay. It took chemists and scientists several attempts to find the perfect structural difference because these RNA errors may lead to dangerous adverse events in the host if they are still picked up by the polymerase and successfully reproduced. It's also interesting to note that remdesivir is an antiviral prodrug that is not activated until metabolized by the body and then serves as a nucleoside analog to prevent RNA polymerase from fully replicating viral genetic information. First, masking the highly negative phosphate groups with esters or amides reduces the molecule's overall polarity, letting it cross the plasma membrane into cells. This can be seen in the protide piece in the bottom right portion of the screen. Second, to be recognized by polymerases, the drug must be phosphorylated in order to resemble a normal nucleotide triphosphate. This can be seen in the upper left-hand corner in the intracellular portion of remdesivir's transformation. We can see this change in synthesis occurring as a protecting group, known as a McGuigan protide, delivers the nucleotide analogs into cells. And here, from its initial inactive state when infused into a patient until it's metabolized in the body and converted in its active state as a nucleoside, remdesivir then reaches its potential to interact and stop the novel coronavirus.
So far, we've gone over the history of remdesivir, as well as how it functions in the body and as a drug. Now we move on to the structure and some of the structural features of our molecule. This is a 2D representation of remdesivir. As you can see, it is a very complicated structure. However, at this point, we are able to identify many of the functional groups that make up the molecule, including an ester, an amine, a phenyl, 2 syn alcohols, an ether, a nitrile, and this nitrogen ring structure called pyrrolotriazinamine. The 2 hydrogens on the alcohols create hydrogen bonds. The hydrogen on the alcohol on the left creates a hydrogen bond with the nitrogen on the ring structure, and the hydrogen on the right alcohol creates a hydrogen bond with the oxygen of the alcohol on the left. This is a little bit difficult to visualize when we're only looking at the 2D model, but as we know by now, molecules like this are not flat the way they appear in this model. They're actually 3D, and the shape that our molecule remdesivir takes is folded, which we will explore more in the next slide. So this is the 3D model of remdesivir. As you can see, it is a lot easier to make sense of why the hydrogen bond is created between the nitrogen and the hydrogen of the alcohol. The positively charged hydrogen on the alcohol is very close to the negatively charged nitrogen on the ring, which we can only really see when we're looking at the 3D model. Now, it's a little bit difficult to see the rest of the model when we're only looking at an image, so we created a video that helps you better visualize the 3D model. So this is our 3D model. As you can see, we've got that phenyl group sticking out at the end, and then the phosphorus where the molecule begins to fold. You can also see those hydrogen bonds.
Hello everyone, and thank you for taking the time watching this exciting organic chemistry project called Remdesivir. As you have seen so far on the previous slides, remdesivir structure has many important functional groups. Remdesivir is a prodrug of an adenosine triphosphate called ATP analog, is from a hydroxyl group of 5-carbon ribose, which can be phosphorylated. Remdesivir is a nucleoside analog that works by blocking the RNA polymerase that coronaviruses and related RNA viruses need to replicate their genes and proliferate in the host. So here we have our references. We have been using important software for our molecule structure and drawing. On behalf of our team, I wanted to thank Dr. Mayer for her support throughout this project. And another big thanks to Valley for supporting us and really made this project possible. And the last but not the least, I want to thank our team for getting on this project early with organizing and editing through the project. And finally, a special thanks and best of luck to you all from Baby Messiah. Thank you all.